The overall goal of this procedure is to create a dynamic microfluidic-based co-culture platform to emulate the intraperitoneal environment with the cellular and mechanical components where ovarian cancer metastasis occurs.
By using this microfluidics platform, it helps to answer the key questions in the cancer biology field, such as the microenvironment of peritoneal dissemination.
The main advantage of this technique is that the 3D co-culture, under precisely controlled fluid flow, better mimics the in vivo intercellular communication between ovarian cancer cells and mesothelial cells that occurs in the peritoneal cavity.
After designing and drawing the microfluidic channel pattern and having it produced according to the text protocol, spin coat a layer of SU8-2075 photoresist onto the silicon wafer with a spinning speed of 1800 RPM. Bake the photoresist-coated wafer directly onto a hot plate at 65 degrees Celsius for 5 minutes, and then at 95 degrees Celsius for 25 minutes to remove excess solvent. Spin coat a second layer of SU8-2075 photoresist onto the wafer for a final thickness of 250 micrometers. Place the photoresist-coated wafer directly on a hot plate at 65 degrees Celsius for 7 minutes, and then at 95 degrees Celsius for 30 minutes, before slowly cooling the wafer to room temperature. Next, align and put the photomask directly on top of the wafer. Then expose it to UV light for 20 seconds to cross.